Today's the topic is the plant tissue culture. So very broad and interesting subject related with agriculture, agri-biotechnology, biotechnology, other biology, applied sciences. This is a very important science in the biology and botany. Plant tissue culture. What is the tissue culture? Plant tissue culture is a collection of techniques used to maintain or grow plant cell tissues or organ under sterile condition and a nutrient culture media of known composition. Plant tissue culture is widely used to produce clone of a plant in a method known as micropropagation. You know that. What is the tissue culture? At the laboratory level, a cell grow into a plant is called tissue culture generally. Plant cells have a capability that is totopotency. Totopotency means every cell gives a new plant at the laboratory tissue culture. It's a culture method on laboratory. So the only plant cells have this capability. That's why this ability is called totopotency. Animal cells doesn't have totopotency. If we cultured a perfect various, for example, we culture a skin cell that gives only skin. If we culture heart tissue cells, that gives only heart tissue. But in the plant, you can take any cell and cultured in the laboratory that raises a new plant. This capability is called tissue culture. Advantage of plant tissue cultures: the production of exact copies of plants that produce particularly good flowers, fruits. So, what is the first advantage of plant tissue culture? The production of exact copies of plants. If you want a good plant, we have so many type of plants. Some plants, fruits are very small, taste is sour. Some plants, fruits are very big, the taste is very sweety. We need that plants. So that time we take the cells from that our desired characters plant cell takes and cultured in the laboratory level and production of that type of plant. This is the good advantages in plant tissue culture. To quickly produce mature plants. So in the method of plant tissue culture, we produce very in time to produce mature plants. The production of multiples of plants in absence of the seeds. Some of the plants like Musa paradisiaca, banana tree, it do not possess seeds, it's not bearing seeds. How we can take the seedlings? So these type of absent of seeds plants, we can take tissue culture, we produce a number of plants for agriculture purpose. The regeneration of whole plants from plant cells that have been genetically modified. So another advantage is if we want to genetically modify a plant in the molecular level, in the cellular level, very easy through tissue culture. The production of plants in sterile containers that allow that to be moved with greatly reduced chances of transmitting of diseases, pests and pathogens. So another important benefit through tissue culture is at the time of culture it is very optimic conditions, very controlled condition we do that culture. That's why the invade of insects, virus, bacteria we can prevent to the plant. This is the major advantage in the tissue culture. The production of plants from seeds that otherwise have very low chances of germinating and growing, that is orchids and nepenthes. Some plants, orchids and nepenthes, are the nepenthes seeds are germination percentage very low. These type of plants very useful for tissue culture to produce new plants. to clear particular plants of viral and other infections and quickly multiply these plants as clean stock for horticulture and agriculture. So, in agriculture sector or horticulture sector, virus-free plants produced through tissue culture. Advantage of plant tissue culture: one, availability of raw material. Very easy availability of raw material, variation supplies and quality, patent right, easy purification of the compound, modification of chemical structure, disease free and desired product, crop improvement. These are the main advantages in the tissue culture. The raw material very easily available to use in tissue culture. At the same time, variation in supplies and quality. Through tissue culture, the plant varieties in variation and quality improves also. Patent rights. Through tissue culture, we produced many type of plants. We got patent also. Easy purification of the compounds. The secondary motable purification is very easy through tissue culture produce. Modification of chemical structure very easy through tissue culture. Disease free and desired product of plants producing is very easy through tissue culture. Crop improvement very high through the tissue culture producing plant. So explain. What is the explain? The part which one used in tissue culture, the part of plant which is used for tissue culture, that part of plant is called explant. The explant is either stem, leaf, whatever it may be. We can take desired part, that is the explant. Any part of the plant taken out and grown in test tube under sterile condition in special nutrient media is called explant. So we take any part, as a flower part or leaf part or stem part or branch part or root part, whatever it may be, we can take for tissue culture. That part is called explant. So method of plant tissue culture. Plant tissue culture includes two major methods. One is the type of in vitro growth callus and suspension cultures. Second one, type of explant: single cell culture, shoot and root culture, somatic embryo culture, meristem culture, anther culture and applied production, protoplast culture and somatic hybridization, embryo culture, ovule culture, ovary culture, etc. We can see the type of explant. Conditions. There are three important aspects in vitro culture. One is the nutrient medium, second is the aseptic condition, third is aeration of the tissue. These are the three major important aspects when we go to in vitro culture. So, one is the nutrient medium. Nutrient medium is essential for the culturing of the part of the plant explant. Medium depends upon the type of plant tissue or cell used for culture. Generally, nutrient medium consists of inorganic salt, organic salt, both micro-macro elements. At the same time, carbon source, usually sucrose we take as a carbon source, sometimes glucose also. Vitamins also very useful in the plant tissue culture. Example, nicotinic acid, thiamine, pyridoxine. These vitamins are included in the nutrient media of tissue culture. At the same time, amino acids, some important amino acid, arginine is used for the tissue culture nutrient medium. At the growth regulators, auxins, cytokines are included in the nutrient medium. At the same time, the pH of the nutrient medium always maintained at the neutral pH, that is optimum pH, that is the 5.7 is very important. This is the nutrient medium conditions. And next, aseptic conditions. When we go to the tissue culture process, the aseptic condition is very important. We can do everything: the preparation of nutrition medium, inoculation of the explant, incubation of the explant. These all are only did in the aseptic condition. So because it is the very delicate things. Delicate things. That's why we maintain aseptic condition. Nutrient medium contains sugar, which increase growth of microbes. That's why we can maintain aseptic condition. These microbes compete with growing tissue and finally kill it. So always microbes utilize the sucrose of nutrient medium. It is completed the cultures tissue. So that's why we always the culture environment is aseptic weather. It is important to maintain aseptic condition. Sterilize is very important to stop the growth of microorganisms. We can do sterilized every glassware, everything medium or whatever we explained, everyone used in tissue culture, that is will be to sterilized. Aeration of the tissue. Proper aeration of the culture tissue is also an important aspect of culture technique. It is achieved by occasionally stirring the medium by stirring or any automatic shaker. It is the aeration is the most important. Due to the aeration, the growth of the explant is very well. If the aeration is locked, the medium at the same time the explant is spoiled. So types of plant tissue culture. Number one, callus culture. In callus culture, cell division in explant form a callus. Callus is irregular, unorganized and undifferentiated mass of activity dividing cells. Darkness and solid medium held by agar stimulates callus formation. The medium contains the auxins and benzyl aminofurin, that is the cytokinin. Both are growth regulators. These elements very important for the callus culture. This stimulates cell division in explant. Callus is obtained within 2 to 3 weeks. Callus culture and subculture. You do the map how that we take the liquid medium. Suspension culture, liquid medium takes suspension culture. The cells slumps on the medium. The cells gives callus. Then scholars change to other medium, developing the callus. So it is most important, callus development, initiation of the callus and suspension culture. At the same time, schematic representation of subculturing. You see that after sufficient time of callus growth, the same medium following changes will occur. Depletion of nutrient in the medium, gradual loss of water, accumulation of metabolic toxin. Hence, for maintenance of growth in callus, it is necessary to subculture the callus. Subculture should be repeated after 4-5 days. As started earlier, cells derived from a single cell. Cells derived from a single cell through mitosis constitute of clone. At the process of obtaining clone called cloning, asexual progenies of single individual make up a clone. There are two popular techniques for single cell culture. Root tips of the lateral roots are sterilized, excised and transferred to fresh medium. The lateral roots continue to grow and provide several roots. After 7 days that are used to initiate stock or experiment. Thus the root material derived from a single radical. It is very important thing in the tissue culture of root tip culture. Such genetically uniform root cultures are referred to as clone of isolated roots. Next, leaves culture. Leaf culture is also one of the important culture in the tissue culture. Leaves may be detached from shoot surface, sterilized and placed in the healthy condition for long period. After sterilized, growth rate in the culture depends on the stage of maturity and excitation. Young leaves have more growth potential than the mature leaves. Shoot tip culture. Excited shoot tips of many plants which can be cultured on relatively simple nutrient media. This media must contain growth hormones and will often form roots and develop into the whole plants. Complex flower culture. Flower culture is one of the most important culture in the tissue culture. We can take 2 days after the pollination. The flowers are taken for tissue culture, excised and sterilized by immersion 5% calcium hydrochloride, washed with sterilized water. Then transfer this to culture tubes containing an agar medium. Fruits which developed are smaller than their natural counterpart. Size can be increased by supplementing the medium with appropriate combination of growth hormones. Anther culture is the another important. Young flower buds are removed from the plant and surface sterilized. The anther are then excised and transferred to an appropriate nutrient medium. The plantlets are formed after 4 to 5 weeks of the inoculation when plantlets are produced from the single anther. Pollen culture, another important culture. Pollen culture, it is used for production of applied plants. Pollen grains are removed from the anther. The first step. Second step, anthers are placed in 5 ml liquid medium in petri dish. Petri dish containing the pollen grains in the culture media are sealed with pharofilm and incubate 28 degrees Celsius in dark for 14 days. 3 to 4 weeks may be required to obtain applied plantlets. It is very useful to produce applied plants. Applied plants useful the genetic experiments and produced few genetic lines. Application of tissue culture. Many applications have the tissue culture. The one first is the tissue culture is used to conserve rare space in the forest. A plant breeder may use tissue culture to screen cells rather than plants. Large scale growth of plant cells in liquid culture. A source of secondary products like recombinant proteins used as biopharmaceutical to cross distantly related species by protoplast fusion. Help in crop improvement, creation of additional genetic variation, that is the somoclonal variation, selection of plant resistant to taxon virus, suitable for breeding of tree species, production of many plants that are a clone to each other, that is called micropropagation, production of disease-free plants. Tissue culture have advantage of time saver. It is very easy to, there is no high technical, there is no require for high technical skills. With the help of the root culture, plants are ready to grow which save time. Thank you. This is the tissue culture, types of tissue culture. We can move next class, micropropagation. Okay, thank you very much, students.